Hello, I'm Ms. Hernandez, and our next student is Matthew Cedillo, and this is our class today, and he'll be doing his presentation.
Hello, my name is Matthew Cedillo, and today I'll be presenting genetic cloning, and specifically the pros and cons of it. I believe this is important because genetic cloning is a up-and-coming scientific field that will be becoming bigger and bigger as time goes on. Potentially within our lifetime, we will see genetic cloning used worldwide.
Continue. It's fine.
All right. Well, in order to understand genetic cloning, we need to know the process of it, how it works. Now, there's two main types of genetic cloning. One of them is called somatic cell nuclear transfer. The other one's a little bit more obscure, it's still being studied. But for today, we'll be focusing on somatic cell nuclear transfer. It basically means that there's two cells, a female ovum and a somatic cell. And when we take the egg cell, we take out the nucleus from the egg cell and we replace it with the nucleus of the ovum. And once they're cloned, we put it into a surrogate mother, which is basically a third cell, which creates a clone cell. And once we have that clone cell, there are two other forms of genetic cloning, which is called reproductive cloning and therapeutic cloning. The reproductive cloning would be the most typical type of cloning, where it's just we're creating a clone of the cell, and the therapeutic cloning would be we're creating more of the specific type of cell. Therapeutic cloning would be used in the medical field as cloning liver cells, heart cells, and within the scientific community, there are those that believe that genetic cloning can help humanity in a plethora of ways. And one of these advantages would be medical advantages. With using somatic cell nuclear transfer in 2001, we were able to clone monkeys, which, as you may or may not know, monkeys are the closest relatives to humans. So that means that if we're able to clone monkeys, we'll be able to experiment on them in order to find a cure for diseases, and also try to find a cure for cancer. And another benefit of genetic cloning would be the resurrection of species in animals and people. Imagine one day later on in life you'll be able to shake the hand of Albert Einstein, or maybe see a woolly mammoth in the flesh. If we're able to find tissue of extinct species, we're able to take the DNA from that tissue and compare it to the closest living relative to the animal, and we're able to once we find the differences between the two, we're able to modify the living relative's DNA in order to resemble the extinct animal's DNA, in order to make clones of the extinct animal. An example of this would be the woolly mammoth. And if we were able to find a tissue of a woolly mammoth, the closest living relative to it would be the elephant. So we'd be able to compare the DNA of the two animals in order to create clones of the woolly mammoth from the DNA of the elephant. And this procedure would also apply to humans if we're able to find tissue samples of notable people such as Albert Einstein or Nikola Tesla. And with all this said, that's pretty much all the positives of genetic cloning. And genetic cloning is not without its downsides, however. And many people believe that the positives of genetic cloning pale in comparison to the negatives that it could bring, not only to the medical field but to society. The disadvantages of genetic cloning, one possible disadvantage is the medical issues that it can bring forth. Using genetic cloning in sciences, in medical field, would be a double-edged sword, because, as I said, genetic cloning is an up-and-coming study, and we don't know the possible outcomes of all the possible outcomes of genetic cloning, which could lead to us creating a cell mutation, a malignant cell mutation that could ultimately bring a lot of loss, a lot of population loss, specifically in the animal that we're studying. And if that gets to humans, then that could bring the end of humanity. Some scientists theorize, like John Harris of the National Institute of Health. The reintroduction, another possible disadvantages would be as of right now, genetic cloning is really trial and error. The first animal that was ever cloned was Dolly the sheep in 1996, and she was the only sheep to be brought forth with a stable pregnancy out of 276 cell fusions. So out of 277 possible sheep, she was the only one to actually be born. So the success rate is really low as of right now. And another possible disadvantage of genetic cloning, which is the more obvious one, would be the ethical and societal issues that it could bring forth. What would the people in the church think if one day we cloned a past pope? We brought back from the dead a pope? Or what would society as a whole think if we were able to reintroduce people into our world? It leaves us to answer a lot of difficult questions as an entire society. Should we spend time and resources into developing genetic cloning, or should we perhaps those resources are better off spent in other areas such as world hunger and climate change? And possibly the bigger question is, who are we to play God? Who are we to play God and create life through non-natural means, through bioengineering, and warp the natural process of nature and twist it to our own needs? The creation of human clones solely for cell lines would, from a philosophical point of view, be an obvious contradiction to the principle expressed by Immanuel Kant, that of human dignity, said John Harris, British philosopher and bioethics, that takes part in the University of Cambridge in the UK. Now, with all this said, by informing you of the possible outcomes of genetic cloning, I hope you understand the arguments for and against genetic cloning. Should we continue research on genetic cloning? Does genetic cloning have the potential to do all the things that scientists propose, or is it a dead-end science project? With all the information presented, make your own informed opinion on genetic cloning, because if we let the study of genetic cloning continue, the era of cloning will be upon us, whether you like it or not. Thank you for your time. This is my presentation on genetic cloning.